This study analyzed two of the deadliest types of cancer that exist, brain cancer and pancreatic cancer. These two cancer types afflict a lot of Americans each year, and unfortunately, in the vast majority of cases, they're not curable. The analyses that we did used, in addition to sequencing analyses, comprehensive analyses of copy number changes. These identify regions of the genome that are gained or are lost, and by combining these analyses together with those that identify point mutation or subtle nucleotide alterations, we are able to get a complete picture of the genetic alterations that occur in these two tumor types in a way that wasn't previously possible, either by looking with one technology alone, or if one had just looked at a subset of genes. The third discovery from these studies that I wanted to highlight was the discovery of a particular gene called IDH1, which was mutated in a high fraction of brain cancers. This gene, which is mutated in about 12% of glioblastomas, which is the type of brain cancer we analyzed, has mutations that appear to occur more frequently in younger individuals. Nearly 50% of patients under age 35 have mutations in IDH1, and it appears that IDH1 may identify a particular subclass of patients that may be different from those that don't have mutations in this gene. It looks like the mutations in this particular gene occur at one particular amino acid residue, and therefore it looks like they dramatically affect the protein function. So in the future, it's possible that by virtue of identifying patients with mutations in IDH1, one may be able to identify patients that may respond differently to different therapies, or for which different therapeutic regimens may be specifically designed. It was quite surprising to find mutations in IDH1. This was a gene that really had never been previously implicated in any tumor type, never been previously implicated, of course, in glioblastomas, and never before identified as either mutated or altered in other ways in other cancers. And I think in a way it validates the benefit of doing a global analysis or a comprehensive analysis of human cancers using the different approaches that I mentioned initially and using an analysis that looks really at all the genes. One can't identify surprises if you're not there and prepared to find them. A question many people may be asking is, why did we choose to focus on these two cancer types, pancreatic cancers and brain cancers? And the truth of it is that it was in large part guided by the fact that these tumors affect a large number of patients every year, and very little is known about them at the molecular level, and very little can be done at the clinical level. Both surgery and chemotherapy are employed, but they're really not very effective. So we urgently need new understandings of the molecular mechanisms of these tumors in order to develop new drugs and new therapeutic modalities that can take advantage of these changes.